Great, well, thank you for checking out this video on cell membranes now. And this is sort of a follow-on from the last video where we talked a bit about lipids, and the idea of lipids is very important to how cell membranes form and why they function like they do. And in one particular one, we mentioned phospholipids in the last video, phospholipids. And we'll just quickly refresh this molecule because it's so important to our understanding of cell membranes. If you remember, we've got a head. This consists of glycerol and the phosphate parts of the molecule, and we've also got the fatty acid legs. I'm going to keep calling them that because it's much simpler if we consider just the two parts to it. So the head and the legs. As we talked about, they've got characteristics. We've got chemical or physical characteristics that are really important. The head is it's polarized, and it's got a hydrophilic properties because of that. So it enjoys reacting with water, and water are polar molecules, so they enjoy reacting with molecules that have also got polarity. They've got charge, attracting the molecules to each other. However, the legs, they are non-polar, and this means they don't like interacting with water. They prefer to interact with non-polar solvents, and for this reason they're hydrophobic. So we've got a molecule that is amphipathic. We talked about that in that video as well. So amphipathic. So it's a single molecule, but each end has got different characteristics. One end wants to interact with water, one end enjoys interacting with water, and the other doesn't. It would rather interact with a non-polar solvent. So if we picture this molecule in the cell, in the cell conditions, it would, if we picture it just existing on its own, it would be surrounded by water. Water is the medium that all our cells exist in. So we'd have water all around it. And at the top end, our head is happy with that interaction. The polarity of it means it is able to interact positively with these water molecules. But the end here, the legs, it's not happy with that. It doesn't enjoy that chemical interaction there. So just by the chemical properties of the phospholipid molecule, it doesn't exist singularly. It doesn't exist on its own within the medium that is the water of our cells. So that leads to questions. Well, how are they going to exist? How do they naturally exist within our cells? And there's two sort of theoretical possibilities. The first one is one that's called a micelle, and this it doesn't actually exist commonly in cells, but that's where the molecules all bunch together with each other. So I'll draw all these heads around it. They bunch together and form a little ball. So it just forms a small little ball. We've got all the heads on the outside, which will interact with the water, and on the inside we've got our hydrophobic legs. They're all interacting with each other. They're all non-polar. The legs, so they're happy. They enjoy interacting with each other. They enjoy coming in close contact with each other. Whilst the hydrophilic heads, they've got the water all around them. So that's straightforward enough. But this isn't a very common shape. If we picture it, you can tell just from my drawing, all these legs are rather crowded in there, and just in terms of sort of physical space occupying, it's too crowded in there for this to be a common structure. So instead, they actually more commonly create a different structure for themselves, and that's the bilayer. So I'm sure you're fairly common with this shape, but if we've got molecules, they line up next to each other, and like this. And if they're lining up next to each other, you could say, oh yes, I'm going to stand next to my neighbour, so water can't get in there. I'm going to be the legs are going to be attracted to each other, and the legs can be happy with their chemical position. But you've got all this space at the bottom here where there's going to be water, so you need something there as well. You need more phospholipid legs. So that's what they do. They match up with other phospholipid molecules, with the legs of other phospholipid molecules. Whilst the heads are happy enough, they'll go on the outside. Oh, wrong colour there. They'll go on the outside, and they will interact with the water. They'll interact with the water of the cell, and they'll be happy enough with that. Whereas the legs are very happy because they're interacting with each other. They're interacting with their non-polar phospholipid legs, of their counterpart molecules, and everyone's happy. We can quite easily see from this molecule that, you know, if it was just what I've drawn here, you'd have water here. You'd have, you know, water molecules that be attracted to these legs again that we're talking about here on the side. So this layer, it goes on basically to infinity until it meets, until it joins up with itself. So if we picture, I'll just draw it as a layer here. So we've got the heads on the inside, and we've got the legs all here. It's going to keep going until it joins up with itself, like this. It's not the best drawing, but it will do, and it will form a continuous layer. I've just drawn this in 2D, but picture this as three-dimensional. It's going to go in all directions until it meets up with itself. And we can see what we've done here. We've created a compartment. We've got water in the middle here, and we've got water surrounding it. But they're separate compartments because the bilayer, it doesn't want to have ends like this where the legs would be exposed to the surrounding water. It wants to only pair up with other phospholipid legs, and that way it forms a complete ring. And the consequence of that is you've got compartments. You've got water that is separated from other water by a complete membrane, and that is what the cells use bilayers for. It's for creating compartments. It is for allowing the cells to control the conditions within the compartments, because they've got, you know, because separate compartments are formed. We're going to talk in the next videos about how cells can control the conditions within the different compartments, but the bilayers are there to just create the compartments first. Why is that important? Why compartments matter? Well, the properties of the phospholipids and the properties of the bilayer are also important, because these are, well, to put quite simply, they're highly impermeable to ions, and to most polar molecules as well, actually. This wrong. It's not the word there. Most polar molecules, and so we've said that the first property of phospholipids is that they naturally form this bilayer structure, which forms compartments. But also these compartments are separated by a layer that has its own properties. It's highly impermeable to ions and most polar molecules. The only exception to this, which I think it's most important to highlight at this time, is that water is the exception to this. So whilst other polar molecules and ions, so sodium, potassium, have great difficulty permeating this membrane, water is not. It is a polar molecule, yes, but it doesn't have any difficulty, or very little difficulty, in traversing this membrane. Why? Well, it's got a very low molecular weight. It exists in a very high concentration. Remember, basically the entire medium of the cells is water based, and it doesn't have a complete charge. So you know, it does have some polarity to it, but it doesn't have a complete charge. So it doesn't have as great a difficulty passing through the membrane. What are the other important properties that we want to talk about at this stage? Well, the first one is that, as we've talked about here, it wants to exist as a complete ring. It doesn't want to have gaps in it. It doesn't want to be open to exploring contact with water, and that gives it the property that these membranes are actually they're actually self-sealing. So they will, if broken, for instance, we'll talk about you know, how vesicles form. In fact, I'll draw a bit now. I'll draw an example of how this happens. So for the picture, we've got a membrane here. So we've got the outside component, we've got the inside, like this, and there's something. There's vesicle formation going on, as we'll talk about here. So first we've got a bend in it like this. Bend in it, sort of colour again. Draw the inside, and then it breaks off. So that's 1, 2, 3, and it breaks off. So we've got break there. We've got our component of the membrane here. Then, as we can see here, we've got the hydrophobic compartments of the membrane exposed to water here, and as I mentioned, it doesn't like that. So what's going to happen is they're going to find a different format for them to be in, a different situation for them to be in, and they're going to seal off. So top here, we're going to have these going to seal off, and this part of the membrane is also going to seal off with itself. And as you can see here, this allows cell membranes to break off from each other to form vesicles here. As we picture, we've got a little vesicle at the bottom here that's grown from the cell membrane itself, and it also allows them to merge with each other. This process can happen in reverse. You can have a vesicle that starts here and it breaks and it blends with its with the membrane to form a larger membrane, and that's how membranes grow. Actually, vesicles are added to the membranes in the same way that they're taken away from it. And this self-sealing, self-healing property allows the cells to function in many different ways. We picture the different cell organelles. We're just talking about the main external cell membrane, but the different organelles are all made up of the same bilayer, and that allows them different components to break off and interact with each other. The final important property that makes these cell membranes so useful is that this occurs spontaneously, all through the chemical properties of the phospholipid molecules themselves. The main driver is the hydrophobic interactions. So it's mainly hydrophobic. So the hydrophobic legs trying to escape the water molecules that surround it. But there's also a positive attraction between the different phospholipid legs. They're drawn to each other, whilst the positive, the heads, the polar heads, are attracted to each other, or attracted to the water molecules that surround them. So it's driven entirely by the chemical properties, the chemical forces that are resulted from this amphipathic amphipathic structure that phospholipids exhibit, and it's formed the compartments that the cell needs to function. And we're going to talk about how, yes, we've got a bilayer now, we've got a layer, but this isn't the end of it, and the cell uses a huge number of different pores in this, different proteins embedded within this bilayer, within this membrane, to carry out a lot of functions, and we'll talk about that in the next video. So before we finish, do a summary of what we've talked about. So we know that for membranes, a lot of their characteristics is based on this amphipathic qualities of the phospholipid molecule, and this having of a hydrophilic and hydrophobic ends, and that causes them to naturally form phospholipid bilayers. And these bilayers, just by the very nature, they form compartments. So they form different cell compartments, which is the main reason they're so important to cells. It allows compartmentalization. They have some very important properties that the cell also needs. They are highly impermeable to ions and most polar molecules, with water being the exception to that. They form spontaneously, as we talked about at the start. They form spontaneously based on the chemical properties of the phospholipid molecule, and they are self-sealing, able to form new structures and incorporate other structures in a constant moving fashion. And all these properties are very important to allowing this to develop its shape, really develop a cell to be different from the external environment, and that's why cell membranes are so important.